Hi everyone, this is our lab for basic animal tissues. It's a great lab and it's going to really put you into your role as a biologist. So let's get into it. This lab is looking at the major tissue types, and there's only 4 of them for animals in the animal kingdom, invertebrates specifically, and it's also going to look at the subcategories of those 4 major types as well. It might seem challenging to think about all the different types of tissues that you're going to see, but we're going to break this down in the mantra of form follows function. So, since form follows function, we're going to study the function of these tissues first. And in studying the function of those tissues, we're going to reverse engineer them and figure out their structure. It makes a lot of sense. They're very well designed. So let's get into their function, and then we'll get into their structure. First of all, what is a tissue? You've done a lot of work in your lab this semester on cells, and that is because we were leading up to groups of cells collaborating to form a tissue. That's where we're at now. Tissues and groups of tissues, different types of tissues, will form organs, like a liver, and then groups of organs can form organ systems, like your intestinal tract. In terms of the tissues that we're looking at, all of these tissues can be grouped just into 4 categories. We're going to be going over epithelial tissues first, then connective tissues, muscle tissues, and nervous tissues. Think about epithelial tissues. You might have known that your epidermis is your skin, so you might have a good feeling for what the epithelium might be. If you need some extra information, you can take it from the Greek prefixes and their words, because most biology is from Greek. For instance, their prefix for a covering upon something is epi. Just think upon or epi, right? Go together. And the Greek word for epite was theia, and that heat was associated, obviously, with secretion, so that you can think about epithelial tissue as being a sheet that lays upon an organ that is probably involved with secretion. Again, form follows function. That's the function. This epithelial tissue is covering an organ that is probably involved with secretion. The function of that covering is to act as a barrier or an external layer over that organ in order to prevent mechanical injury, pathogens and their injury, fluid loss, and dehydration. Another function would be to let that sheet that is laying over the organ interface with the environment, so the organ doesn't have to interface with the environment. This epithelial tissue will do that instead, and it will moderate secretions, absorptions, excretions of that organ. The organ has enough to do. Let's think about epithelial tissue and my friend the wolf. Again, we talked about the epithelial tissue being a barrier. Well, the biggest organ any animal has is their outer skin. So we can think about epithelial tissue and the epidermis, like you know, as the outer skin of our wolf. Also, we see this arrow here leading to the lining of his mouth. Well, epithelial tissue inside of his mouth will certainly prevent mechanical injuries if he happens to chomp on a bone, or invasion by pathogens if he ate bad meat, or fluid loss as he pants. So the linings of the mouth, also the lining of the anus and the vagina, are all epithelial tissues. When we talk about the function of these epithelial tissues interfacing with the environment, we are talking about lining or covering organs that usually manage gases or liquids. And we were talking about secretory organs, so the liquids certainly fit in there. But there's a lot of epithelial tissue that is involved with the respiratory tract, because we're not only managing gases in the respiratory tract, we're also managing water vapor and dehydration as well. So epithelial tissue is related to mucosal tissues and the lung tissues that we have. We'll go into that in just a second. Also, our wolf has epithelial tissue in his intestines. Those are certainly involved with secretion and absorption, and in his kidneys as well. That makes sense. And we forgot about his salivary glands in his mouth. So you can see that epithelial tissue is probably one of the most prevalent tissues that you'll find in the animal kingdom. And let's explore it a little bit more. When we think about those two functions, barriers and interaction with the environment, we can think about what type of form this tissue will take. Well, if you have a barrier or a sheet, you want tissues that have uniform cell shapes. You don't want different size or different shape cells. They won't fit together well to form a good sheet. You also want these cells to be tightly packed with junctions that really hold the cells together to prevent that secretion from leaking. It would be a great idea if those barrier tissues were primarily avascular. In other words, they don't have a blood supply. That way, when they get mechanical injuries from wounds that they ate, there's not a lot of blood that's involved with those injuries at the epidermis or at the epithelial tissue. We want to conserve our blood. That's why many cuts that you get really don't bleed. That's a primarily avascular tissue in your epidermis. At the same time, think about the function there. These tissues get a lot of wear and tear. So we want them to be able to sustain rapid growth and replacement. Form follows function. You're going to have avascular tissues that have a uniform shape and that are tightly packed. One surface is going to be exposed to the environment so that the organ doesn't have to do that. And one surface is probably going to be anchored to connective tissue that is sitting right on top of the organ. And this one surface will be anchored to a basement membrane that will go right onto the connective tissue. We'll get into that. Let's look at the form following the function. We talked about the layers and the tightly packed layers that we would assume would follow for epithelial tissue. And we could have very simple epithelial tissue with just one layer of cells, or we could really pack in this epithelial tissue and have it stratified. You have to look at the function of each of these tissues. For instance, this flat tissue would be great at getting absorb or taking in or diffusing gases right into an organ beneath it. It has a lot of surface area and it's a very simple tissue. So this area right here has an apical surface like the apex or the tip or the top connected to a basal or basement membrane. Very simple. The same way, if you needed more cushioning, like you needed more cushioning on the neck of your wolf, so its mama can actually pick up her puppies by the neck, you might need some more cushioning there. You have the same basement or basal layer, but you might have a lot of cells tightly packed in a stratified tissue with an apical or top surface that is nice and flat. So this is a way to look at form follows function by number of layers. You can also look at the shape of the cells that are very regularly shaped in epithelial tissue. Squamous cells are flat, great for diffusion and getting gases into lung tissue. You can remember that word squamous because you get think of squished cells. Cuboidal cells would be great for kidneys that have, of course, a lot of secretory function, and the cuboidal cells can hold a lot of liquid. The columnar-shaped cells might be involved in anchoring down a cilia that would help in, especially in the trachea, where there is cilia and mucus in there. So you could anchor some long cells in a columnar cell, a cell like a cilia in there, and that might be a taller cell for epithelial tissue. Epithelial tissue is named by looking at either simple or stratified. The number of layers is the first word of the name, and the shape is the second word of the name. Let's go into that a little bit more. For instance, when we go back to look at a bit of our work with our wolf, we are coming from one cell that we were looking at, this simpler columnar cell, one cell, and then we're trying to get to the point where we can look at tissues or groups of cells that are draw a tissue, and then we're going to try to get to the point where we can see a slide and relate what we know about the function to the form that we're seeing in front of us and name that tissue. We'll get there. Go back to our wolf, and to go back to looking at drawing our tissue, we can take our flat, typical squamous cells, add on a basement membrane, and call this a simple squamous epithelial tissue. It is very well known that most animals and their lung organs have an enormous amount of simple squamous epithelial tissue. Let's look at that from a drawing of a tissue to a slide of a tissue. This is of the air sacs of the lungs. This is actually an air sac. The air sac is bounded by one layer of squamous cells that is diffusing gas back and forth along that very simple basal membrane and squamous cell tissue. If you think about the function of exchanging gas, you're going to want maximal surface area. You're going to want a flat squamous squished cell and a flat nucleus there as well. If you think about a cuboidal epithelial tissue, that cuboidal epithelial tissue could easily be in a kidney tubule. Kidneys are associated with an enormous amount of secretion. When your function is to secrete urine, you're going to want a cell that can hold a maximum volume and really hold a lot of bulk in there. A cuboidal cell and a cuboidal epithelial tissue really does take care of that. And at the same time, you have a lot of space there for a large nucleus. When we look at columnar epithelial tissue, that can be involved in the intestinal tract. Think about the function there. You're absorbing nutrients. You don't know what's in that nutrient. Maybe it's something you might want to check out and keep away from your nucleus before it got all the way down into the cell. So a tall cell that keeps its nucleus down at near the basal membrane might provide some good protection for this nuclear material in here, having a tall cell